Hello students. So all of you have just given your NEET UG 2023. I'm pretty sure you all must be anxious to know whether the question is correct for you or it was incorrect for you. So in this lecture, we are basically having a discussion of the paper of NEET UG 2023 and I will be dealing with the botany part of it. So we will go through each and every question of botany. We'll see what is the answer. I'll explain you the answer of the question and then you can check whether it's correct or incorrect for you. So let's start with the discussion of NEET UG 2023 botany paper. So if you see the question number 101, here we can say that given below are two statements, right? Statement 1 says the force generated by transpiration can lift a xylem-sized column of water over 130 meters height. So here they are talking about the xylem column which is 130 meter height, which is a correct statement. Okay, so this is true. This statement is true. Now if you see the second statement, transpiration cools leaf surface. So transpiration is where the evaporation of the water will take place. So when the evaporation of the water will occur, so cooling effect is given by to the plant. So transpiration cools the leaf surface sometimes 10 to 15 degrees by evaporative cooling. So the temperature reduces by 10 to 15 degrees when evaporation cooling which occur due to transpiration. So this statement is also correct. Okay, so you can see in the light of the above statement, choose the most appropriate answer from the options given below. So we see the first option, both statement 1 and statement 2 are correct. So we can see both the statements are true. So these are correct statements. Option 2 says both statement 1 and statement 2 are incorrect. So we have just seen that both the statements are correct. Option 3 says statement 1 is correct but statement 2 is incorrect. And the option 4 says statement 1 is incorrect but statement 2 are correct. So what is the correct answer? Correct answer is both the statement 1 and statement 2 are correct. Both the statement, that is the force generated by transpiration, can lift a xylem-sized column of water over 130 meters height is true, as well as transpiration cools leaf surface sometimes 10 to 15 degrees by the evaporative cooling. So both the statement is correct. Let's move on to the next question. Now the next question says that again given below are two statements. One is labeled as assertion, that is A, and the other is labeled as reason, that is R. Now let's read the assertion. Assertion says the first stage of gametophyte. Now gametophyte is the haploid part of the life cycle. So the first stage of the gametophyte in the life cycle of moss. Moss is a bryophyte. So bryophyte is where the gametophytic stage, that is the haploid phase is the dominant stage. So in mosses, the gametophyte is of two types. One is your protonema, which is filamentous green structure, and the other stage is your leafy gametophyte. Okay. So when the spore is released from the capsule, the spores will fall on the substratum and it will germinate to form the protonema, that is the filamentous structure. Okay, so the first protonema will develop. On the protonema, the buds will develop, which will later lead to the formation of your leafy gametophyte. So the first stage of the gametophyte in the life cycle of moss is protonema stage, which is again a true statement. This is true. Second, the reason, if you see, protonema develops directly from spore, which is correct. The spores which are released from the capsule, that will fall on the substratum and it will develop into the protonema. So it directly develops from the protonema produced in the capsule, which is true. This protonema will later give rise to the leafy gametophyte. So this is also true, and this is also the correct reason. Why? Because first the protonema will be formed from the spore and then the leafy gametophyte is formed, and directly from the spore the protonema is forming. That's why it is the first stage of your life cycle in the moss, the gametophytic life cycle in your moss. So both the statement is correct and the reason is explaining the assertion as well, right? So if we see the first option, both A and R are correct, and R is the correct explanation of A, which is correct. Second option, both A and R are correct, but R is not the correct explanation of A. Third option, A is correct, but the R is not correct. And the fourth option, A is not correct, but the R is correct. So what is the answer for this? Answer will be this. Both A and R is correct, and R is also explaining the assertion. Reason is explaining the assertion. So let's move on to the next question. Now, unequivocal proof that DNA is the genetic material was proposed by the Hershey and the Chase experiment. Okay, Hershey and Chase experiment, which worked upon the bacteriophage, where the protein and the sulfur, the sulfur was labeled, and in the DNA the phosphorus was labeled. So that experiment was used to give an unequivocal proof that DNA is the genetic material. So the DNA is a genetic material, unequivocal proof was first proposed by, first option if you say Frederick Griffith, second option if you say Alfred Hershey and Martha Chase, third option Avery, McLeod and McCarthy, and the fourth option is Wilkins and Franklin's. So which is the correct answer? The correct answer will be your Alfred Hershey and Martha Chase. So they performed the Hershey and Chase experiment, which gave the unequivocal proof that DNA is the genetic material. Next question. Now the thickness of ozone in a column of air in the atmosphere is measured in terms of Dobson unit. So Dobson unit is the unit for measuring the thickness of ozone present in the atmosphere. So we know in the stratosphere the ozone layer is present, which is responsible for protecting the earth from your harmful UV radiations. So the thickness of the ozone is measured in terms of Dobson. So if there is a thinning of the ozone, the Dobson will decrease, the layer of ozone will decrease, and harmful UV radiation will be able to cross the atmosphere and reach the earth's surface and will cause harmful effect in the living organism. So the unit which is used to measure the ozone layer is your Dobson unit. So next question. In a tissue culture experiment, the leaf mesophyll cell. Now leaf mesophyll cells are what? These are your permanent cells. Okay, these are your permanent cells, differentiated cells. So these are put in a culture medium to form callus. Callus is what? Callus is your undifferentiated, or we can say meristematic. Meristematic cells. Okay, so from a permanent cell, that is from the differentiated cells, undifferentiated cells are getting formed. So from a differentiated cell, that is nothing but your permanent cell, undifferentiated. That is your meristematic cell is getting formed. So what is this process called? The process is called as de-differentiation. De-differentiation. So differentiation is a process where from a meristematic tissue, a permanent tissue which will be formed. But from a permanent tissue, when a meristematic tissue is formed, that process is called as de-differentiation. So here you can see the leaf mesophyll cells, which is your differentiated permanent cells, they are getting converted into a callus, which is your undifferentiated cells, a meristematic tissue. So what is this process called as? This phenomena will be called as your de-differentiation. The phenomena will be called as de-differentiation. Let's see the next question. So a large, colorful, fragrant flowers with nectar, right, are seen in what? First option, insect pollinated plants. Second option, bird pollinated plants. Third option, bat pollinated plants. And the fourth option is your wind pollinated plants. So the correct answer for this will be your insect pollinated plants. large, colorful, fragrant flowers. So this is just to attract the insects for the pollination. So this flower, which are large, colorful, fragrant and have nectar, these are your insect pollinated plants. Next question. Frequency of recombination between gene pairs on the same chromosome as a measure of the distance between the genes to map their position on the chromosome was first time used by first Thomas Hunt Morgan, Sutton and Boveri, Alfred Stuyvesant and the Henking. So answer for this will be your Alfred Stuyvesant, right? He was the one who basically used it for the first time. So he was the one who used the distance between the gene as the equivalent to the recombination frequency between the gene, the crossing over which is between the gene. So the number of crossovers or the frequency of the crossovers is directly proportional to the distance between the genes. And this phenomenon was used to make the genetic map and to place the different genes on the chromosomes. Next question. Which of the following stages of meiosis? Okay, they are talking about the meiosis. So which of the following stages of meiosis involved in the division of the centromere? Now division of the centromere means if this is the chromosome having the two chromatids, the centromere will divide like this, division will take place and the two chromosomes will be formed. Two chromosomes like this will be formed when the division of the centromere will occur. Now this will occur in case of your anaphase 2. Okay. So metaphase 1, there the chromosome, the two chromatids of the chromosome does not get separated, the centromere will not split. Metaphase 2 also, there will be no splitting of the centromere. Anaphase 2 is where the splitting of the centromere will occur and a chromosome having two chromatids will get split and two chromosomes will be formed. So doubling of the chromosome will occur during the anaphase 2, right? Let's move to the next question. What is the role of RNA polymerase 3 in the process of transcription? So they are talking about the RNA polymerase 3 in the process of transcription in eukaryotes. Now you know in eukaryotes 3 RNA polymerase is there, RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. RNA polymerase 1 is responsible for the synthesis of 28S rRNA, then 5.8S rRNA as well as 18S rRNA. The RNA polymerase 2 is responsible for the formation of mRNA and the RNA polymerase 3 is responsible for the formation of tRNA, 5S RNA and snRNA, small nuclear RNA. So first transcription of rRNA, that is 28S, 18S and 5.8S, that is RNA polymerase 1. So this is done with the help of RNA polymerase 1. Second, transcription of tRNA, that is 5S RNA as well as snRNA, that is done with polymerase 3, RNA polymerase 3. Transcription for the precursor of mRNA, this is done by your RNA polymerase 2. And transcription of only snRNA is not only snRNA, tRNA as well as 5S rRNA is also involved in by RNA polymerase 3 during the process of transcription. So correct answer for this will be your option 2, transcription of tRNA, 5S rRNA as well as snRNA. Let's move to the next question. Among the 4 evil quadrants, which one is considered the most important cause driving extinction of the species? So the most important quadrant will be your habitat loss and fragmentation. When the habitat is not there for the organisms to survive, so obviously they will lead to extinction of that organism. So the most important, so here the key is most important. So the most important is your habitat loss as well as your fragmentation. So first option, habitat loss and fragmentation. Second, over exploitation for the economic gain. Third, alien species invasions, and the fourth, co-extinctions. So the main and the most important reason or the most important evil quadrant will be your first, that is your habitat loss and fragmentation. Next, given below are two statements. First statement, endark and exarch are the terms often used for describing the position of secondary xylem. No, it is used for describing the primary xylem, because primary xylem is divided into metaxylem as well as protoxylem. So depending upon the position of protoxylem and the metaxylem, the xylem is divided as endark or exarch. Okay, so it is not the secondary xylem, it is the primary xylem which is involved. Second statement, exarch condition is the most common feature in the root system, which is true. So this statement is true, but the first statement is false. So in the light of the above statement, choose the correct answer from the options given below. First option, both statement 1 and 2 are true, which is false. Second option, both statement 1 and statement 2 are false, which is again incorrect. Statement 1 is correct, but statement 2 is false. And the fourth option, statement 1 is incorrect and the statement 2 is correct. That is your correct answer which is there, right? So here it should be primary xylem, then the statement would have been correct. Let's see the next question. Okay, axile placentation is observed in mustard, cucumber and primrose. Second, China rose, bean and lupin, tomato, dianthus and pea. And the fourth option will be China rose, petunia as well as lemon. So axile placentation is where the entire ovule is divided into multiple chambers, multiple locular, and the ovules are present in the center of the locule. Okay, center of the locule. So you can see axile placentation will be like this. This is how the locule will be there, multiple locule, and here you will have the ovules that will be attached. This is your axile placentation, which is seen in China rose, petunia as well as lemon. Okay, so the correct answer for this will be your fourth option. Next, expressed sequence tag, that is ESTs, refer to. Now it refer to first of all genes which are expressed as RNA, which is correct. Because express sequence tag only take into account the genes which will be coding for protein. So it is the one which get transcribed from the RNA, and then from the RNA translation will occur and the proteins will be formed. So if we see the next option, all genes that are expressed as proteins, all genes whether expressed or unexpressed, and certain important expressed genes. So the correct answer will be all genes that are expressed as RNA. That is your expressed sequence tags. Okay? Okay. So next question. What is the function of tassels in the corn cob? Now tassels are nothing but it is your feathery stigma. Okay, hairy stigma. So it is responsible to capture the pollen grain so that the pollination will be happening and then fertilization will take place. So the function of tassels in the corn cob is to attract insects, that is no. To trap pollen grain, that is true. To disperse the pollen grain and to protect the seeds. So the correct answer will be what? Second option, to trap the pollen grains, right? Let's move to the next question. Spraying of which of the following phytohormone on juvenile conifers help in hastening the maturity period? So it fastens the maturity period that leads to early seed development. So the development of the seed will be faster. First, indole 3-butyric acid, which is nothing but your auxin. Second, gibberellic acid. Third, zeatin. And the fourth is your abscisic acid. Okay, so the correct answer for this will be your gibberellic acid. Gibberellic acid is responsible for the shortening of the maturity period for your juvenile conifers. Okay, next question. Now in the equation, GPP minus R is equal to NPP. GPP is gross primary productivity. NPP is net primary productivity, and the R is what? R is your respiratory losses which occurs in the organism. So R is what? Photosynthetically active radiation, which is not. inspiratory quotient, which is not, respiratory loss, right, and the reproductive allocation, which is again incorrect. So R here is nothing but your respiratory losses. So GPP minus the respiratory loss will be equal to your NPP, right? Let's move to the next question. Now, family Fabaceae differ from Solanaceae. Okay, Fabaceae is nothing but your legumes. Solanaceae is the potato family. And Liliaceae, that is onion, garlic, lily. With respect to stamen, that means they're talking about your male reproductive structure, androecium. Pick out the characteristics specific to family Fabaceae. So something which is specific to Fabaceae. It is there in Fabaceae but is not found in Solanaceae and Liliaceae. So Diadelphus and Dithecus. Now Diadelphus condition is a characteristic feature of your Fabaceae family which is not there in your Liliaceae as well as Solanaceae. Okay. So this Polyadelphous condition is seen in case of your citrus plant, so it is not there in Fabaceae. Epipetalous condition is seen in case of your Solanaceae but not there in Fabaceae. Monoadelphous condition is seen in case of Malvaceae, again not in Fabaceae. Monothecus, again it is there in case of your Malvaceae family. Epiphyllus is there in Liliaceae family. So the correct answer for this will be your Diadelphus as well as Dithecus anther. Diadelphus and Dithecus is only there in Fabaceae but not there in Solanaceae as well as your Liliaceae family. Right? Next question. In angiosperm, haploid means N, diploid means 2N and triploid is your 3N. Structures of a fertilized embryo sac sequentially are. Now first is your synergid. Synergid will be your N. Primary endosperm nuclei will be your 3N and the zygote will be your 2N. So this option is wrong. Antipodal will be your N. Synergid is N and the primary endosperm nucleus will be your 3N. So again this option is wrong. Synergid is N. Zygote is 2N, primary endosperm nucleus is right, so this option is correct, 3N. Synergid is N, antipodal is N, polar nuclei is again N, so again this option is negated. So the correct answer will be what? Third, synergids, zygote as well as pen, primary endosperm nucleus. Okay. Next question. Which hormone promotes internode petiole elongation in deep water sea? So here the key point is deep water sea. Internodal elongation will occur through gibberellin also. But deep water sea will be your ethylene. Ethylene will be the one which is occurring in deep water sea, due to which the rice plants stand erect. So Ga is gibberellic acid, kinetin is nothing but your cytokinin and T4D is your auxin. So the correct answer for this will be your ethylene. Okay, ethylene hormone. Next, given below are the two statements. One is labelled as assertion and the other is labelled as reason. Assertion: late wood has fewer xylary element with the narrow vessel, which is true. This statement is true. Why? Because cambium is less active in winter. So in the winter the requirement of water is less, so the cambium will be less active, due to which it will form the autumn wood or the late wood. And in the autumn wood and the late wood, the number of xylums, vessels as well as the trichies will be less, as well as they were narrow, because the requirement of water will be less. Okay. So hence the cambium is less active in winter, which is also true, and the correct reason also. It is explaining why the few xylary element and the narrow vessels will be formed. So if we see the first option, both A and R are true and R is the correct explanation, which is correct. Both A and R is true, but R is not the correct explanation, it is explaining the assertion. A is true and R is false, and the fourth is A is false but the R is true. So the correct answer will be your first option. Both A and R is true and R is the correct explanation of A. Let's see the next question. Given below are two statements, one labelled as A and the other labelled as R, assertion and reason. First, ATP is used in two-step of glycolysis, which is true. ATP is used in the payoff phase of glycolysis at two steps. first step as well as the third step. So first step where glucose converts to glucose 6-phosphate, and the next third step is where fructose 6-phosphate converts to fructose 1,6-bisphosphate. So in both the cases, ATP will be used as a phosphate donor. So if you see, the first ATP is used for converting glucose into glucose 6-phosphate, which is true, and the second is used for the conversion of fructose to fructose 6 diphosphate is again true. So both A and R is true, and R is also the correct explanation of A. So both A and R is true, and R is also the correct explanation of A. Next question. Identify the correct statements. Detrivose performs fragmentation, which is true. The humus is further degraded by some microbes during mineralization, which is again true. Water soluble inorganic nutrients go deep down into the soil and get precipitated by a process called leaching, which is true. Next, the detritus food chain begins with living organism is false. The detritus food chain begin with detritus. It begin with detritus. And the last, earthworm break down detritus into small process by the process called as catabolism, which is wrong. The process is called as fragmentation. The process is called as fragmentation. So how many correct options are given below? A, B and C. So only A, B and C is correct. D and E is incorrect. Right? Let's see the next option. Next question. The process of appearance of recombination nodule. Now, recombination nodule is formed during crossing over, exchange of genetic material between the non-sister chromatid of your homologous chromosomes. So, the recombination nodule will occur in which substage of prophase 1? That will be a packetine. Why? Because during the packetine only the crossing over will take place. Next. How many ATP and NADPH2 are required for the synthesis of one molecule of glucose during Calvin cycle? So, they are asking for complete Calvin cycle, right? So, it will be your 18 ATP and 12 NADPH. So, we can see during the reduction step, 2 ATP and 2 ATP and 2 NADPH is used during the reduction step. During the regeneration step, it uses 1 ATP. Okay, this is for 1 CO2 fixation. So, for 1 glucose, it will require 6 CO2 fixation. So, it will be 6 into 3 ATP plus 6 into 2 NADPH2. So, it will be your 18 ATP plus 12 NADPH2. Okay. So, 18 ATP and 12 NADPH2 will be used for one molecule of glucose during the Calvin cycle. Next, the phenomena of pleiotropy. Pleiotropy means a single gene is coding for multiple phenotypic effects. So, a single gene code for multiple phenotypes. That is called as pleiotropy. So, first option, presence of several alleles of a single gene controlling a single crossover, that is not there. Presence of 2 alleles, each of the 2 genes controlling a single trait. A single gene affecting multiple phenotypic expression, and more than 2 genes affecting a single character. So, the last option is polygenic inheritance. So the correct answer for this will be a third option, a single gene affecting multiple phenotypic expression. Next, the reaction center PS2 has absorption maxima as 680 nanometer. So PS1 will be your P700 and PS2 will be P680. Okay, P700 and P680. So PS2 will be your P680. Next question. Which micronutrient is required for splitting of water molecule during the photosynthesis? So that will be your manganese. For splitting of water, we have manganese, chloride and calcium. Okay, so out of the three, manganese is involved in splitting of water during photosynthesis. Next, movement and accumulation of ions across the membrane whenever is against their concentration gradient, that means energy will be used for transporting the molecules from low concentration to high concentration. So this is done during active transport. So this will be during active transport. During facilitated diffusion, down the concentration gradient it will be moving. Osmosis and passive, again down the concentration gradient it will be moving. Against the concentration gradient will be explained by your active transport. Next, among eukaryotes, replication of the DNA. Now DNA replication will occur in the S phase. S phase of the cell cycle, S phase of your interphase is where the DNA replication will take place. Next, the historic convention on biological diversity, that is the Earth Summit, was held in Rio de Janeiro in the year of 1992. So in 1992, the Earth Summit happened in case of Rio. Identify the pair of heterosporous pteridophyte will be your Selaginella as well as Salvinia. Lycopodium, Coleoterm as well as Equisetum, all these are homosporous. Heterosporous is Selaginella as well as Salvinia. Now list 1 and list 2. So we have to match the columns. M phase is your equatorial equational division. G2 phase is where the proteins will be synthesized. Quiescent stage will be your inactive phase, and the G1 phase is the interval between the mitosis and the initiation of your DNA replication, that is your S phase. So you can see A will be 4. Okay, so this two options can be negated. B will be 1, so this option. C will be your 2 and D will be your 3. So option third. Again match the following. Now iron is used as an activator of catalase. Zinc is used as synthesis of oxygen. Molybdenum is used a component of nitrate reductase, and boron is used for cell elongation as well as differentiation. So you can say A will be your 3. So these two options gets negated. B will be 1. So this is your answer. This gets negated. So option third. Again match the following. Cohesion. Cohesion is the mutual attraction among the water columns. So force of attraction between the similar molecule is called as cohesion. Adhesion, it is the attraction towards the polar surfaces present on the xylem, the attraction of the water to the polar surfaces of the xylem. Surface tension, it will be more attraction in the liquid phase, and guttation will be the loss of water in the liquid phase. So A we can see will be your 2. So A is 2. So this gets negated as well as this gets negated. B will be 4. So again this option. C will be 1 and the D will be third. Right. So option 1. Next, How many different protein does the ribosome consist of? Ribosome consists of 80 different types of proteins, which is there 80 different types of proteins are there in the ribosomes. Again, list oxidative decarboxylation occurs by pyruvate dehydrogenase, where pyruvate converts into acetyl-CoA and the acetyl-CoA will enter into the Krebs cycle. Glycolysis is also called as EMP pathway, Abenden, Meyerhoff and Parnas pathway, depending upon the name of the scientist who proposed it. Oxidative phosphorylation will be your electron transport system, and tricarboxylic acid will be your citrate synthetase, right? So A will be 2, so this 2 option gets negated. B will be 4, C will be 3. So this option, C will be 3. So option 4 in this case. Next, again match the following. Mutualism. Mutualism is where both the species are getting benefited. So both A and B will be positive. Commensalism is where one is benefited, the other is neither affected nor benefited. So it will be your first option. Amensalism is where one is harmed, but the other is neither affected nor harmed. And the parasitism is where one is benefited, the other will be harmed. So A we can see 4, so this option gets negated. B we can see 1, so these 2 options gets negated and the correct answer will be 1. C will be your 2 and the D will be your 3, right? Next, given below are 2 statements. Gauss's competitive exclusion principle states that two closely related species competing for the same resources cannot coexist indefinitely. They cannot coexist indefinitely, and competitively inferior will be eliminated eventually, which is a correct statement. In general, carnivores are more adversely affected by competition than herbivores. This is wrong. Herbivores are more affected than the carnivore. So first statement is correct, but the second statement is wrong. So first statement is correct and the second statement is false. Next. Given are the 2 statements, assertion and reason. So first statement, a flower is defined as a modified shoot, which is true, wherein the shoot apical meristem changes to floral meristem, that is true. The internode of the shoot gets condensed to produce different floral appendages laterally at successive nodes instead of leaves. So again that is true and also the correct reason. That is the reason why the shoot apical meristem get converted to the floral meristem and the leaves will get converted into the floral wreaths. So both the statements are correct and R is also the correct explanation of A. So both A and R are true and R is the correct explanation of A. Next, which of the following statement is true about Klinefelter syndrome? So the disorder was first described by Langdon Down, which is wrong. The Down syndrome was done. Such an individual has overall masculine development. However, the feminine development is also expressed, which is true. The affected individual is short-statured, that is not there. Physical, psychomotor and mental development is retarded, again not there. Such individuals are sterile, which is true. So option B and E. B and E statements are correct. Okay, the other statements are wrong. Next question. Now, which of the following statement is not correct? That means they are talking about the incorrect statements. The microorganisms involved in biodegradation of the organic matter in a sewage polluted water body consume a lot of oxygen, causing the death of the aquatic organism. That is true. Algal bloom caused by excessive organic matter in the water improves water quality and promotes fisheries. That is false. It is harmful for the water bodies as well as the aquatic life. Water hyacinth grows abundantly in the eutrophic water bodies and leads to an imbalance in the ecosystem dynamics of the water body, which is true. And the amount of some toxic substance of industrial wastewater increases in the organism at successive trophic level, which is true, which is nothing but your biomagnification. So this is true. So the only incorrect statement will be your second. So the answer will be your second statement. Next, which of the following combination is required for chemiosmotic theory, chemiosmosis, which occurs during the ATP synthesis in mitochondria as well as in case of your plastids, chloroplast. So for chemiosmosis, first of all, ATP synthetase will be required, a proton pump will be required, a membrane will be required, and a proton gradient will be required. So a membrane, proton pump, proton gradient, and ATP synthetase. Second option, if you see, membrane, proton pump, proton gradient, NADP synthetase is not required. Proton pump, electron gradient is not required. So correct answer will be your first option. Membrane, proton pump, proton gradient, as well as your ATP synthetase. Next, select the correct statement. Identify the correct statements. Lenticels are lens-shaped opening permitting the exchange of gases, which is true. Bark formed early in a season is called as hard bark, which is wrong. It is called as the soft bark. Bark is a technical term, is wrong. Bark is a non-technical term that refers to all the tissues exterior to the vascular cambium. Bark refers to periderm and secondary phloem, which is true. Phelogen is a single layered in thickness. Okay, it could be single layered or it could also be multi-layered. So the correct answer from the option given below. So the B and C is incorrect. So B is incorrect, C is incorrect, B is incorrect. So option will be your second, A and D. A and D are your correct statement which is there. Next question. The two statements, assertion and reason, and the first assertion, in gymnosperms, the pollen grains are released from the microsporangium and carried by air current, which is true. Anemophily will occur. Anemophily will occur. Wind pollination will occur in case of your gymnosperms. Reason, the air current carry the pollen grains to the mouth of the archegonia where the male gametes are discharged and the pollen tube is not formed, which is incorrect. Pollen tube is formed. The male gametes in case of gymnosperms, mostly in case of gymnosperms, is non-motile. So they require a pollen tube for carrying it to the female gamete. So the reason is false, but the assertion is true. The reason is false, but the assertion is true. So option third. Next. The broad palm with single palm crease is visible in the person suffering from Down syndrome. So this is the most common symptom of the person suffering from Down's syndrome. Next. Which of the following are not considered as a part of the endomembrane system? Endomembrane system has 4 cell organelles: lysosomes, endoplasmic reticulum, Golgi apparatus, as well as your vacuole. So mitochondria, chloroplast, peroxisomes are not the part of your endomembrane system. So the most appropriate answer will be your A, C and E. They are not the part of the endomembrane system. Next, gene A will be coding for trans-acetylase, Y will be coding for permease, I will be coding for repressor protein, and the Z will be coding for beta-galactosidase. So A will be 2, so this option and this option gets negated. B will be 3, so this option gets negated and this is your correct answer. C will be your 4 and the D will be your first. Next. Which of the following function is carried out by cytoskeleton? Cytoskeleton is your microtubules and microfilaments, which are the major components of your flagella as well as cilia. So both the cytoskeleton in a cell will be responsible for motility. Motility will be involved when it comes to your cytoskeleton in a cell. Which of the following statement is correct? Eutrophication refers to increase in domestic sewage and wastewater in the lakes. Biofagnification refers to increase in concentration of toxicant at successive trophic level, which is true. Presence of large amount of nutrients in the water restricts algal bloom, and algal bloom decreases fish mortality. Both all these statements are wrong. So the correct statement will be second. Biomagnification means the amount of toxic waste will keep on increasing as we move throughout the different trophic levels in a food chain. Next, two statements are given below. In prokaryotes, the positively charged DNA is wrong. The DNA is negatively charged. So positively charged DNA is held with some negatively charged protein, that is false, in a region called nucleoid. In eukaryotes, the negatively charged DNA is wrapped around the positively charged histone proteins to form nucleosome, which is correct. So statement 1 is incorrect and the statement 2 will be correct. Next, a leopard and a lion in a forest or grassland will be competition. A cuckoo laying egg in crow's nest will be brood parasitism. Fungi and root of higher plant in mycorrhizae will be mutualism, and the cattle, ergot and cattle in a field will be your commensalism. So A will be 1, B will be 2, B will be 2, C will be so A will be 1, B will be 2, C will be 3 and D will be 4. So let's see the next question. Given below are the two statements. Electrostatic precipitator is most widely used in thermal power plant, which is true. Electrostatic precipitator in thermal power plant remove ionizing radiation, which is not true. It is used for removing the particulate matter. So statement 1 is correct, but the statement 2 is incorrect. Statement 1 is correct and the statement 2 is incorrect. Next, RNA mutates at a faster rate, which is true. Statement 2, viruses having RNA genome and a shorter lifespan mutate and evolve faster, which will also be true. So both statement 1 and 2 will be true. Both statement 1 and 2 are correct. Next, which of the following symbol represent mating between relatives in human pedigree analysis? That will be this symbol. This symbol is used to represent the mating between relatives. Mating between the relatives in human pedigree analysis. Next, first, logistic growth is when the resources are limited. So, limited resource availability conditions. Exponential will be unlimited resources will be available. Expanding age pyramid where the percentage individual of the pre-reproductive is largest, followed by your reproductive and the post-reproductive. So this will be this, and the stable will be the percent of individual pre-reproductive and the reproductive groups are the same. So A will be 2, B will be 1. So this option, this gets negated, this gets negated and this gets negated. So option 1. So given below the two statements, during G2 phase, the cell cycle during the G0 phase of the cell cycle, the cell is metabolically inactive, which is wrong. It is metabolically active. The centrosome undergoes duplication during S phase of the interphase, which is true. So the first statement is wrong and the second statement is correct. So the first statement is incorrect, but the second statement is correct. Next, the tetrad formation is seen during the leptotene stage, which is false. Tetrad is seen during the pachytene stage, or we can also say during the zygotene stage. During anaphase, the centromere splits and the chromatid separates, which is true. Terminalization will occur during pachytene, that is false. Terminalization will occur during dikinesis. Nucleolus, Golgi and ER are reformed during the telophase, which is true. Crossing over takes place between the sister chromatid, that is wrong. Non-sister chromatid of your homologous chromosomes. So choose the correct answer from the options given below. Which statements are correct? So option B and D. So both B and D are correct. Only B and D will be correct. Right? Next. Which of the following is the sequence corresponding to the coding strand? So, if suppose this is the DNA, 5' to 3', 3' to 5', and the RNA is formed from 5' to 3'. So, this is your RNA. Okay. This will be your template strand and this will be your coding strand. So, first of all, coding strand is from 5' to 3'. So, this as well as this option gets negated, right? Secondly, the DNA molecule is there in a coding strand. So, their uracil will not be present. What will be present will be your thymine. Okay. So, this will be your answer. First of all, it has to be 5' to 3'. Okay. Secondly, the coding strand will be exactly same as the RNA molecule, mRNA molecule, only the uracil will be replaced with your thymine. So, if you see this is the sequence of the coding strand will be 5' A, T, C, G, A, T, C, G, A, T, C, G, A, T, like that it will be there. Uracil will be replaced with your thymine. So, option third. So, this was the botany paper of NEET UG 2023. So, I hope you were able to attempt all the botany questions correctly and have answered all the questions in botany correctly, and you don't have any doubt in the paper now that why that is the answer for the question, and I hope you got the correct answer key for the botany section of the NEET paper. Thank you.